بسم الله الرحمن الرحيم. به نستعين. اللهم علمنا عنك يا رب، فهمنا عنك يا رب. Third day, sequence, alignment and foundation of bioinformatics. Sequence alignment is a fundamental concept in bioinformatics. What we do in this sequence alignment, we compare a DNA or RNA or a protein sequence. We compare it with what? With a known already sequence. Okay, so we uncover hidden similarity, revealing clue about shared function, evolutionary relationship, and conserved regions. This understanding empowers us to make a critical discovery across a diverse field, from drug development to evolutionary biology. Let's understand what is sequence alignment. Sequence alignment is a process of arranging two or more sequences. The sequence can be DNA, RNA, or protein to identify region of similarity. Okay, so we can bear a sequence that is unknown. Okay, we discovered through our lab scientific research to already known DNA sequence in our database. What are the types of sequence alignment? We can pairwise alignment compare two sequence, and we can compare multiple sequence alignment, comparing through 3 or more sequence simultaneously. Align method. Align method include global and local. Global alignment, we align sequence end to end, from the start to end, identifying the optimal alignment across their entire length. Example include Needleman-Wunsch algorithm. Okay, there is another type called a local alignment. It focuses on finding a region of similarity within the sequence, even if they are not perfectly aligned across the whole length. Okay, like Smith-Waterman algorithm, it is a key example. In what we use the sequence alignment as an application? Number 1, identifying homologous genes. Identify a homologous gene. Sequence alignment help to identify gene, protein with a shared ancestry across different species. We can predict function of a known gene. How we can do this? Number 1, we compare this unknown sequence to a known one in our database, and the known one we already know its function, so we can predict the function of the uncharacterized gene. Okay, we having a known gene, we having a known sequence. This unknown sequence is already we found a similarity with a known gene, and this known part we have similarity with it having already known function, so we can imply from that this unknown gene having this similar function. We can study evolutionary relationship. Sequence alignment help to understand evolutionary relationship by analyzing how closely related the sequence. Okay, we can design primer for PCR. Sequence alignment is crucial for designing primer that accurately amplify a specific DNA sequence. What are tools we can use in this sequence alignment? So here we are talking about tools. There is what is called BLAST. BLAST, it is abbreviation for Basic Local Alignment Search Tool. It identifies regions of local similarity between sequences. Another tool is called Clusteral Omega. It is a popular tool. It performs multiple sequence alignment. Here is an example you can apply to use one of these tools. Okay, let's practice on BLAST. Okay, number one, you will access to the BLAST website. Okay, you can input your query sequence. This sequence can be DNA, RNA, or protein. Then you select appropriate database for your search. There is many database: nucleotide sequence, protein database. Then you can run the BLAST search. This is a broad guideline, but I advise you in this. Use ChatGPT. Ask him that please guide me step by step in practice in using BLAST. Okay, he will give you the website link, will give you example of gene, will guide you step by step. It is really an interesting journey to practice what you learn. So use ChatGPT in guiding you step by step. Okay. After we use the BLAST tool to make the sequence alignment, I need to interpret results. Okay, there is a key concept. He will introduce it to us. You will see what is called e-value. Okay, what is meant by e-value? It indicates the likelihood of finding a match by chance. Lower value of the e-value, it indicates a great significance. Okay, identity, percentage of identical residue between query sequence, this unknown sequence, and the database match. Score, numerical score, it reflects similarity between my query sequence and the database match. So these concepts will help me in future to interpret my BLAST result. Okay. Beyond the basics of alignment, okay, beyond the basic, we can align multiple sequences. Multiple alignment, we will align 3 or more sequences simultaneously. What am I aim? Reveal the conserved regions. Application, in what I use this multiple alignment. If you interested in phylogenetic analysis, which is a concept, we will talk about it in detail. If you need to understand or predict protein structure, how this sequence of amino acids that form that protein will fold and form protein structure, okay, and this structure I can imply from it its function. And functional study, you're trying to understand the function of unknown protein, unknown sequence of a DNA. What are the tools that you use? Cluster omega, muscle, tea, coffee. سبحانك اللهم وبحمدك، أشهد أن لا إله إلا أنت، أستغفرك وأتوب إليك. Thank you.